Well, good afternoon. My name is Gene Giacomelli, and I'm the director of the Controlled Environment Agriculture Center here at the University of Arizona. And for all those in the room, welcome. And for all those out there on the web, welcome as well. We appreciate you being here for our first Covering Environments, part of our new seminar series that Rafi Gruner has put together for us. We really appreciate all the work that he's done. There will be a series of seminars once a month throughout the school year that, by the way, we just had the opportunity to survive the first week, and with many more to come. Again, I want to welcome you here. I'd like to mention that this is a special year, particularly for the Controlled Environment Agriculture Center. And although I don't see her in the room, she's probably in the greenhouses working right now, Dr. Pat Rohrbaugh will be retiring this year. And I want to keep bringing that up. We're going to be calling this the year of the Rohrbaugh. Yes, you should chuckle a little bit. The idea is to keep her in our memory, and remember that as every day passes, every event that occurs, this is Pat's last opportunity to be a part of it in the position she stays in right now. Next June 30th, she will retire, and it will become a major challenge for us to replace her teaching the class the basic controlled environment hydroponic greenhouse course. We'll look forward to a year of many opportunities to mention this. I'd like to introduce Rafi Gruner. He's a designated campus colleague for the Controlled Environment Agriculture Center, which means he volunteers his time and he does things like organizing a seminar. So, Rafi, if you would please
introduce the speaker, okay. Get this going. I would just like to say that I'm really heartened to see that virtually every seat is taken. I used to run a seminar series in physiology, my old department, and I always had close to heart attacks just before the seminar, because if you have a party and nobody shows, that's really terrible. So thank you all for coming. It's wonderful to see you. I think the seminar series is going to be a great step intellectually for everybody who attends and everybody who is on the website, because we're going to sort of expand topics, and today is one good example of that, to do not only what is directly related to CAA, but also things that tie into it. And we're going to have some very interesting topics. I would just like to mention that the next one after this, next month, is going to be given by Ed Horton, who is the owner and CEO of Urban, the very high-tech installation in Irvine, California. It's vertical farming using hydroponics, recirculating, and it should be absolutely amazing. But today, we're very fortunate to have Barry, who is a member of the Plant Sciences Department, and Dr. Pryor is one of the gems that I keep discovering at the University of Arizona, even though I've been here over 40 years. And that is that he has special knowledge that is unique, because I didn't know that anybody at the university actually grows mushrooms. And those of us who are older remember mushrooms from the good old days that took you to far out places, but I don't think that's the kind you grow. But anyway, he has a very similar kind of outlook on mushrooms that connect to the human condition, whether it's history, religion, medicine, wars, and everything else. So I'm really excited to hear your story, Barry, and thank you again for giving the inaugural seminar of the SEAC.
All right. Well, thank you. I think I have this. Thank you very much, Ravi, and thank you, Ravi and Jean, for inviting me to seminar series. So today I'm going to talk about mycoculture. And mycoculture, integrating mycoculture into diverse farming systems. And with the concept that by doing this, we're going to be able to close food production and energy loops. When I put together my seminar today, I really wanted to talk about the how and the why, or the why and the how of making this integration. And as I got up to about 80 or 90 or 100 slides, I realized that was way too many slides. And I'm going to have to cut it down. So today I'd like to focus a little bit on the why we want to integrate mycoculture, production of mushrooms, into these farming systems. And maybe, Gene, at a later seminar, will talk about the specifics of how we're exactly going to do this, okay? So I usually don't start my slide with my presentations with the acknowledgements. But mycoculture has been part of my program for only about 2.5 years. And I really have to acknowledge and I'm grateful and inspired by some of the people who helped develop this program, particularly these two students, Lauren Jackson and Parker Evans. Wow, is this a synergistic combination in my lab? Both of these students took a course that I have, Mushrooms, Molds, and Man, and maybe that's where Rafi was referring to, and how I try to integrate mushroom production into humans and human affairs. And Lauren and Parker took this class, and then they joined my lab. And from then on, we just took off running. They were the ones who started me in mushroom cultivation. They were the ones who started developing these programs. Over the course of this time, we've got funding from the Green Fund. I'll talk about this. We developed this program called the MycoCats, and we're funded by the Green Fund. We've been funded for a few years by the Green Fund. We've also recently got some new funding from the Arizona Department of Agriculture, so we are expanding. We are expanding. But the MycoCats is a student-driven organization. We have up to date. We've had approximately 45 interns. And Lauren and Parker have been instrumental in keeping this program alive and growing. And as all professors know, we stand on the shoulder of our students many times. So, and this is the case. So I have to acknowledge them in this program, and probably them for many of these slides as well. I sort of steal the slides from my students periodically. I mentioned the funding, the Green Fund, Arizona Department of Ag. We have a lot of collaborators in this program, particularly Tucson Village Farms. We have some participants of Tucson Village Farms out here. SEAC, of course, my Green Fund, or our Green Fund sponsorship, it includes, or is co-PI is Dr. Cherry Kubota here at SEAC. We've cooperated with the Compost Cats, another Green Fund sponsored sustainable program at the University of Arizona. Facilities management has been instrumental in giving us resources. I have to acknowledge all these people in this effort over the last couple of years. Okay, so I'm going to start this talk by giving you a little bit of a mycological primer. And I've learned from my course that mycology is a subject that a lot of people don't know a lot about. Even though we've taken biology back in high school and college and everything, we learn about bacteria, we learn about viruses, we might even learn about protozoans, but we don't learn much about fungi, which I think is very interesting, okay? So I'm going to give you a little bit of a mycological primer, sort of warm us up and get us all on the same page. What is a fungus? Okay, real quick definition. Achlorophyllus. That's a hard one to say. Achlorophyllus. Filamentous. Reproduced by spores. Absorptive processes. Nutrition obtained through absorptive process. This is a key, okay? Have chitin in their cell walls. These are half-life. These are part of the biology that differentiate fungi from other organisms. What I want to talk about today really is this obtain nutrients through these absorptive processes, because this is what fungi do. This is what is key to fungi. Here's some examples of some fungi, and of course we all know yeasts are fungi too, right? Yeasts are fungi. Not filamentous fungi, but they are fungi. Okay, so if we want to look at the sort of the kingdoms of life, where do fungi fit in? They are neither plants nor animals. They are their own unique kingdom. Okay, and this is an old tree of life. It's our 5-kingdom tree of life. There's really 7 kingdoms right now. But I'm just going to put this up because really, by looking at this, the plants, the fungi, and animals, and looking at their mode of nutrition right here, we can see how this separates, how it fits together really nicely. Plants obtain nutrients from photosynthesis. Animals obtain nutrition from ingesting nutrition, and fungi obtain their nutrition through absorptive processes. They secrete these enzymes, and then they absorb the degradative products from this enzyme degradation. Okay, now we can look at this sort of, excuse me, where is my slide? Okay, and through this absorptive process, fungi play a key role in the environment. Key role in the environment. Ecologically, fungi are the single most important decomposers on the planet, both in natural ecosystems, breaking down wood products, breaking down our grass, our shrubs. And if it wasn't for fungi, we'd be up to our skyscrapers in logs and brush. They would just break down so slowly. But fungi can ramify through these substrates with their hyphae, secrete these enzymes, and break them down into smaller bits, and then bacteria will take it on down the recycling process. But natural products and man-made products, fungi, with their arsenal of degradative enzymes, can break down practically any man-made product we've come up with today. Wood products, textiles, now I'm drawing the blank, textiles, computer disks. They can etch glass, right? They can break down petroleum products. They can break down most of our pesticides as well. And so these mountains of trash that we're generating, we could turn that loose on fungi, and they would be breaking it down into more of a recyclable form, okay? And if you look at this in sort of putting them all together, the whole picture, fungi are the third critical component, and we don't think about that often. They're the third critical component in recycling energy and resources. Normally we think of things as the producers, the plants, photosynthesis. The consumers, the animals, they ingest. But without fungi as the intermediary in there, bringing those resources, bringing that energy back to plants, things slow down. They get recycled, they move it across. And in fact, you could probably just kind of cut animals out of here, and plants and fungi, and plants and fungi, they themselves can form a great cycle of producing energy, producing carbon, and then recycling it back to a form that can be again utilized. So most specifically then, and with that in mind, most specifically, fungi and plants go together like peas in a pod. Fungi are really the greatest promoters and then recyclers of plants. Okay, fungi are the primary colonizers of rock surfaces due to lichens. Once they start breaking down rock surfaces, they form soil. Plants can come in and grow from the soil. Fungi also are the primary symbionts of the plants in which they encourage to grow through their breakdown and the weathering of rocks, as mycorrhizal fungi. Okay? And it is, as some people have suggested, that plants would never have been able to colonize dry land without this symbiotic relationship with fungi through mycorrhizal association. And then, interestingly enough, fungi are the primary pathogens of plants. Not for us. For us, it's bacteria and viruses. But for plants, it's fungi. They're the primary pathogens of plants and then the primary decomposers of plants. Interesting relationship. They both help the plant grow. 90% of all plants have mycorrhizal associations with their roots. They help the plant grow, and they make the plant sick and kill it, and then they recycle it. What a great system. What a great system. Okay? All right. Armillaria, the humongous fungus. Up in Oregon, this one species, one individual of armillaria covers, I don't remember the exact, it's like 5 square miles. It's been thought to be the largest organism on Earth, a single entity. And they know it's single because they can do DNA testing on all the parts, and they know it's a single individual. So you can, the largest organism on Earth, and perhaps the oldest organism on Earth. It might be 10,000 years old, 12,000 years old. Okay? They're phenomenal. And we don't learn this in school. That's the interesting thing. Okay, plants and fungi have an ancient relationship, mycorrhizal relationship with plants and fungi. Okay, and this is a timeline of fungi diversification based upon a DNA clock. And we see that fungi, if you go back and the mycorrhizal fungi often are part of the genus Glomus. And if you follow the clock back here, the Glomus starts radiating in the, how do you pronounce it, Ordovician period, right? Ordovician? I think I learned it out. Ordovician period, half a billion years ago. Half a billion years ago. Now, plants were really colonizing the planet up in the Devonian. But back in the Ordovician period, there's maybe bryophytes crawling out of the ocean. And they were it has been, and due to these findings from fossil records, they found glomeruline fossils dating back to almost 500 million years ago. Already, fungi and plants were associated when plants were no more than just bryophytes on rocks. It's an ancient relationship, very ancient. Okay. Okay, and the fungal-algal relationship, okay, similar to that lichen is a symbiosis between fungi and green algae, or some blue-green algae. In fact, it's my most famous, or my favorite joke, right? I usually don't do jokes, but this one's a good one. Why did the fungus marry the algae? Because they were liking each other. Oh, okay, it's a good one, right? Everybody gets that one too. Okay, so fungi, this is a really ancient relationship, right? And oh, I know. Oh God, comedy. This is Comedy Central now. We didn't sign up for this. All right. So you know when those islands erupt out of the ocean, volcanic islands, the first thing that's on there is our lichen colonizing the rock, making soil. Okay. When global warming runs rampant and everything dries out, the last thing that's going to be there are lichen on the rocks. And then when the sun starts fizzling out and we have turned back into snowball earth and everything gets frozen, the last thing that's going to be there are lichen on the rocks. Okay? And this is almost science, and it's so strange that it's so durable that you think, how could this possibly be of this earth? Well, some people think that life did not evolve on Earth. There's the Panspermia theory, right? That life actually evolved somewhere else and was blown to Earth on meteorites or bits and pieces of rock floating around. And so there's been a series of experiments, particularly the Biopan experiments. 2,000 funders sent them up on a Russian module here. This is the Biopan module. It was packed by the European Space Agency with something like 36 different studies on physiology and chemistry, biophysics, in weightlessness, right? And they had some biology experiments in there as well. They put up some bacteria, they put up some algae, some worms, and they put up lichen, okay? This is, when they came crashing back down to Earth after only 16 days. This is a quick experiment. We've known that bacterial spores can survive in years in space, but only a few days unshielded of solar radiation, right? Green algae, less than 24 hours, even when shielded. Green algae and their fungal symbionts as algae survived over 2 weeks at 90% unshielded. That is one tough organism. And it's interesting that symbiosis imparted a durability to both partners, the phycobiont and the mycobiont. Okay? It's an amazing relationship. All right. So, okay, that's a little bit about my primer. But now the question we have, and what we're always interested in here at SEAC and in the School of Plant Sciences, is, of course, the $7 billion question. You've seen this on many slides, and I think I got this, this was early this morning, okay. 7 billion on the planet. By 2050, we're supposed to have 9 billion. And the pressing question is, how do we feed an increasingly populated world? How are we going to do this with decreasing arable land? This is a challenge. This is a big challenge. And the School of Plant Science is addressing this challenge. It's one of our mandates now. And we do this by focusing on plant breeding, new genotypes, cultural practices, plant protection, biotechnology, and of course, controlled environment systems, right? But what about fungi? Who's talking about fungi here? Can fungi become a substantial and a sustainable food source? That's the key right there. Can be substantial and sustainable. Okay, edible mushrooms. Here's the history of edible mushrooms. And Jean sent me an image earlier today, and I actually had already incorporated it onto my seminar. Earliest records of human use of fungi, of mushrooms, 6,000 years ago. Oldest archaeological evidence of mushroom art in some of these caves in Spain. And here's this, this auroch, these big bisons, and down here are these all little mushrooms painted. And the people who found this cave said, oh, those look a lot like Psilocybe hispanica. Psilocybe, we all know about Psilocybe, right? So at least if you grew up in the 60s, you knew, and the 70s, you knew about Psilocybe, right? Those are the recreational mushrooms, right? Okay. And then they're also key to many religious sects in Central America and the Middle East. They've used these mushrooms for millennia. Okay. 3,000 years ago, Iceman, Otzi the Iceman, remember everybody found Otzi. He wasn't carrying any food, but he was carrying two different kinds of mushrooms, right? Pedopterus betulinus and Fomes homitarius. Okay, Pittoporus is known as the birch polypore. It has all these interesting medicinal properties through this polyporic acid. They're antibacterial. They are anti-helminthic properties. So when you got bad guts, which I'm sure Otzi had many times, right? You eat this mushroom. This is called a tinderbark conch. They use it for fire starting. It also has some antimicrobial properties as well. So Otzi was carrying these things. This was in his toolkit. No food, though. He was just living off the land. But he made sure that was in his toolkit, his medicine kit, all right? And then, of course, we had all kinds of interesting stories. Julia Agrippina kills Emperor Claudius with Amanita. We know all these stories, too, allowing son Nero to ascend to the throne. So all these sort of insidious uses of mushrooms. And I thought it was interesting when I put these together because here we have the mushrooms used in spirituality. Here we have mushrooms used in medicine. And here we have mushrooms used in much more insidious purposes. Okay, interesting use of mushrooms. But what we're interested in, though, is the edible mushrooms. That's what we're going to talk about today. Okay, there's about 2,000 species of edible mushrooms that have been documented. People know they're edible. That's out of probably about 20,000 species of fungi that actually produce a basidiocarp or an ascocarp, a mushroom, right? By the way, only about 40 species are known to be toxic out of all those. And maybe only a handful are acutely toxic. So this whole fear about mushrooms, it's not well. About 60 of them have been cultivated, right? About 10 on a large industrial scale. All the rest of those wonderful 2,000 mushrooms have to be gathered in the wild, and they are gathered in the wild. They've been gathered in the wild for years. It is a multi-billion-dollar industry worldwide, and if you go up to the Seattle, the Portland, or the San Francisco mushroom market, you will see an explosion of mushrooms, of beautiful, wild, harvested mushrooms of all kinds. It's wonderful to go there. Just even if you're not a fungophile, right, a lover of mushrooms, go to the market. It's really wild, all right? I should have put a picture up here. Okay, and right, most the wild harvested mushrooms are considered gourmet mushrooms. These are not just things you can barely eat. These are things that you desire. Okay, the morels. Of course, we know the morels. 45 bucks a pound. They're very expensive. So if you know of a good morel patch, you can go out in an afternoon and collect over 10 pounds of morels. That's pretty good money. So just in an afternoon, 500 bucks. You go home. You're good. Okay, chanterelles are the same. If you know your good chanterelle patch, those good that oak grove that's just bursting with chanterelles. You can find cauliflower mushrooms up on Mount Lemmon. You have to hunt far and wide from this. Spiracis is the genus. Very delicious. And of course, we all know about the truffles. Very expensive. Black truffles, $140 a pound. This is the cheap variety. This is the cheap variety of black truffles. The expensive variety of black truffles, $2,000 a pound. These are from unique groves, unique forests. Okay? Most wild harvested mushrooms cannot be commercially grown. They're strictly mycorrhizal, as are the truffles. You can't grow them in culture. Not yet. Okay. From specific forests, specific genotypes, and they're delicious, $2,000 a pound. This, in fact, is our attempt at artificially cultivating truffles. We plant an oak forest, right? And we seed it with truffles. So at least we're not wandering around crazy in the forest. We actually know where to go. And if you're out there with your truffle pig, right, at least your pig doesn't have to walk too far, right? Okay. You got to save that pig. That pig's very valuable. All right. So some of the wild harvested mushrooms, however, can also be grown commercially, such as some of these meadow mushrooms or some of the wood decay mushrooms. And we grow them on compost or on sterilized substrate. The beginnings of commercial mushroom production started in the 13th century in China, when we had a description and the preparation of 15 mushroom varieties on wood chips and straw. This is the beginning of it, of people trying to cultivate something that was previously uncultivatable and wild harvested. It continued on the cultivation of shiitake mushrooms on logs. In France, 18th century in France, is where we really started in the large-scale commercial production of mushrooms with the cultivation of Agaricus campestris, our common meadow mushroom, and on artificial beds. And this initiated modern commercial mushroom production. Of course, we did it on horse manure and straw compost, and we did it in limestone caves. It was done in limestone caves. Why do you think limestone caves? It had nothing to do with the lighting. Mushrooms grow fine in the light. In the forest, they're growing in light. It has to do with the limestone cave was a controlled environment, right? It's constant temperature, constant humidity. You can grow mushrooms down there. In fact, they had to put lights down in there. Okay, and then in the last century, we started growing oyster mushrooms on sterilized substrate or pasteurized substrate, and this was the first commercially cultivated sort of gourmet mushroom. Okay, mushroom production in the U.S. right now is a $1.5 billion industry and growing. What do we have? 65% is in Pennsylvania. That's very odd, but we'll talk about that in a moment. 15% is in California, and 20 years ago, this was probably 2%. And now another 19% is in other states, including Arizona. This is where we hope to increase. How do we compare the crop value with other crops that are grown in the U.S.? Okay, these are some of our common and some of our most lucrative crops: potatoes, tomatoes, lettuce. Look at the pie slice of mushrooms, almost the size of sweet corn. You know, the same crop value as sweet corn. Sweet corn, we think, oh, we all know sweet corn, but we don't all know mushrooms. But yet, it's a pretty valuable crop. We're using mushrooms a lot, and whether you know it or not, there's mushrooms in many of the products that we eat. Okay? Okay, the champion of this is Agaricus bisporus. This is the common button mushroom. And actually, Agaricus bisporus has been in cultivation long enough that we have many different selections of it. So the white button mushroom, the portobello, are the same species, kind of like broccoli and cauliflower, right? But we've selected for interesting genotypes. And the crimines are also the same thing. We've selected for interesting genotypes. And so we're already selecting for button mushrooms of unique quality. 80%, 70% of the mushrooms produced in this country worldwide are these. These are easy to grow, and they're durable. You can ship them across oceans. That's a big thing. Okay. Is growing mushrooms profitable? This is what we all want to know. Very cost effective. Most of the raw materials are inexpensive. They're agricultural byproducts, right? Straw, poultry waste, horse manure, corn cobs. Many things that are normally just thrown away, or maybe composted. Okay. High yields, generally over 6 pounds of mushrooms per square foot of compost. Pretty good. And if you start pricing that, $3 a pound wholesale, it's $20 a square foot. If you wanted to put it in an acre amount, $800,000 an acre, right? If you grow gourmet mushrooms, which are about $80 a square foot, $3 million an acre. I don't know if cannabis can pull in that sort of money. Perhaps it can, maybe. But what? Not on that. Okay. We're learning about cannabis, right? Okay, it's right next door in Colorado. But remember, though, mushrooms you can vertically grow. You can put shelf after shelf after shelf after shelf. So 1 square foot here adds up to 10 square feet if you put 10 shelves. It's incredibly lucrative. And mushroom market. What are the projections of mushroom cultivation right now? New study by Markets and Markets suggests that the $30 billion worldwide industry right now by 1919 is going to be over $50 billion. That's almost doubling the price. There's not a single other crop with that growth prospect projected in social. And why? Why is this? Why is this? Oh, here's the trend. And we can see it. Here's the trend. 1961, 2012. Oh, where's my colors? I don't have colors. Oh, there they are. Okay. I got these slides from my student. They put all this. Okay. Anyways, so there's China, though. The big surges in China. The big surges in China right now. They're going commercial mushroom production like gangbusters, okay? And then we have, Italy's there. The U.S. is down there. Kind of flat, but it is growing. Okay, Netherlands, Poland, okay. But you can see the trend. It's really growing. Okay. And why is it growing? Why is it growing? Because people are learning. People are learning the amazing properties, the delicious and nutritious properties of mushroom. That's why they're growing. That's why we're learning to become fungophilic. We've been fungophobic for too many centuries. Now we're learning about mushrooms and we're embracing them. Okay, are mushrooms good food? Of course they're good food. Some say mushrooms have no nutritional properties. Oh my gosh, how misinformed were those people? For example, let's look at the B vitamins, for example. Okay, high in B vitamins, riboflavin, niacin, folate, panthic acid, very high in the B vitamins. Some of the minerals as well. Protein content, 3 grams of protein. 3 grams of protein per 100 grams. Let's see, is this 100 grams? Where is it? 84 grams, serving size, 3 grams. Okay, but remember, mushrooms are 90+% water. If you do it on dry weight, it's over 40% dry weight. Let's compare that to some other kind of products, such and we know this, right? Mycoprotein in mushrooms is an incredibly balanced protein combination of all the essential amino acids. Similar in amino acid ratio as that of eggs. It's a complete protein source. First class, high in fiber, no cholesterol, low in fat, no trans fat. It's amazing. Compare that to corn, regular corn, 10% protein, 10% protein, dry weight, or less. Deficient in 3 essential amino acids. Mushrooms are a much better protein source than corn, much better. And the British knew this too. During World War II, when they were sending off all the beef and the chickens and the pigs and everything and the eggs off to the troops, they were concerned about the population in England. And they said, oh, we're going to have a protein deficient population. And they went and actively started investigating new sources of protein, and they came up with this product here called, cutely enough, corn, Q-U-O-N, right, which is a mycoprotein product, 100% Fusarium venetatum. That's not even a mushroom. That's a filamentous fungus, and they grow it in a big bioreactor, and then they drain it and pack it into cakes, and they make little McNuggets or pancakes out of it, and they flavor it up with everything. It's a great protein source. And if you want to try this, go to Whole Foods. You can get it at Whole Foods. It's about the only place in town I see it. And you can get these little corn McNuggets, and they're wonderful. You get a little ranch, and you dip them in ranch or ketchup or salsa. It's wonderful, and it's mycoprotein. Vegans love it, right? No cholesterol. It's wonderful. Okay, this is a good product. Look at this, tacos, corn tacos. Put it in there. You should have that next time, Gene. All right, other strange, okay, so there's a lot of strange fungal foods out there. And some of us have eaten Vegemite or Marmite, and I put this up here just simply because I wanted to show what happens when you take a different kind of fungus and you make a food product out of it. And this is a yeast extract, and we know that yeast are fungi. This is a yeast extract, and it's thick, not much water weight. And look at the nutritional facts of this. It's really strange. Let's focus on here. What do we got? Amount per serving. 16 grams. And it's interesting to say serving size is 2 heaping tablespoons. Oh my gosh, only the British could heap that on. You know, because it's pretty, it says heaping tablespoons, 16 grams. Protein, 9 grams of protein out of 16. Oh, 50%. Huge protein. Look at the B vitamins, thiamine, 670%. And with this serving size of your of vitamin B1, B2, B3, 280%, 560%. Folic acid, I think B9, 180%. B12, 400%. It's incredibly nutritious. People don't think this way, but we are learning, and that's why the mushroom market is growing so much today. Okay. Also, those amazing nutraceuticals. This is where I think we should be eating mushrooms. This is why we should be. Conjugated linoleic acids. Powerful anti-carcinogens, okay? Significant effects on metabolism, lower body weight, increasing lean body mass, which is this metabolic ratio that doctors like to use, lean body mass, increasing lean body mass. Beta-glucans, reduce serum cholesterol, right? Enhance both the innate immune response and the adaptive immune response. Ergothioneine, a natural antioxidant, we find it in meat, we find it in bran. We have a high concentration of that in mushrooms. And this is a darling of the antioxidants now. If you go on markets and see what you pay for people who can produce ergothioneine, it's like $16,000 a kilogram. It's outrageously expensive. The statins, okay, the statins are the wonder, the savior of the pharmaceutical industry in many respects. We all know about lovastatin. That was the first statin. That is such a powerful, where am I? Right here. A powerful cholesterol modulator. Inhibits cholesterol production by the liver, right? What is the new statin they come up with? It's Lipitor. Lipitor is also a statin. It's a derivative of lovastatin. Lovastatin was first found in oyster mushrooms, right? Oyster mushrooms. Lipitor is the single most profitable pharmaceutical worldwide. In 15 years, I think the profits are $120 billion. In the last year alone, over $10 billion. Lipitor, the single big, and that's a cholesterol, anti-cholesterol drug. And the first one came from mushrooms, came from oyster mushrooms. All right. And then, of course, let's see, aromatase, 5-alpha-reductase, these inhibitors. These are hormone inhibitors, okay? Both estrogen and dehydroxytestosterone. They modulate the hormones. And we all know that as you get older and your hormones level are elevated, those are potent carcinogens, right? Or tumor stimulators. Tumor stimulants, I should say that. And by inhibiting these two enzymes, we actually are reducing our chances of developing cancer in many cancer forms. So these are the amazing things. And okay, so these are the big 5 medicinal mushrooms. I'm not going to go into these that much. We have shiitake, maitake. But the point I'm trying to make here is all mushrooms, even the lowly button mushrooms, have these properties. Even the lowly button mushrooms. So you can't say, oh, we need to eat the fancy ones. No, the button mushrooms are good. We all know Dr. Wells from this University Center of Integrated Medicine. He's put together this anti-inflammatory food pyramid about 3 years ago, okay? And I really like this food pyramid because up at the top you have a glass of wine and a bar of chocolate, and I think, he has it right. He really knows what to do here. But right smack in the middle of it, and this is taken off the USDA pyramid, right in the middle of it, cooked Asian mushrooms. Unlimited amounts. And the reason is because they're so low and they have so low calories, okay? Unlimited amounts. And the point is to eat your fungi. But again, I think he's a little wrong here. Eat any mushroom. All mushrooms are good. And I like to tell everybody, if you want to live to be 100, eat your mushrooms. Eat your mushrooms. This is why the mushroom market is skyrocketing right now. Okay, how do we incorporate fungi into our food production systems? Okay, most commercial mushroom production is done in dedicated control environment facilities. Right, here's some examples of some right here. And these are huge production systems for strictly focused on mushrooms. Okay, and mushroom can be grown in a variety of ways, integrated into food production systems in a variety of ways. You can grow them on open beds, portobellos, white button mushrooms. You can grow them in high and very expensive facilities or very poor, kind of third world facilities. You can grow them indoors or outdoors. You can't grow them outdoors in Arizona. You might be able to do them outdoors, an open bed in, say, Portland or Seattle. Maybe. You need the right environment. You need the right environment. You can grow them in bags. You can grow them in jars. In jars is really neat because you can automate the system when you do it in jars. You have machines that go plunk, 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 move things around. They can fill up the jars. They can inoculate the jars. They turn them over. You have machines to do all this. It's really cool to see. You can grow them on hay bales. TVF on your hay bales? No, you can't do it in Arizona. You have to put a bag around it. You can grow them on artificial logs. You just get wood chips and pack them in. You can grow, whoa, stop, okay. You can grow, so artificial logs, shiitake on artificial logs, shiitake on natural logs. You can grow natural logs outside. It was really how they used to grow shiitake. We brought it indoor now. But it's a really interesting operation that you drill holes in the log and you pack it with these little wooden dowels that are impregnated with the shiitake. And then you put it out in a yard and let it colonize for a year. Why would but if you have the land and you got the forest, you can put them out there, let's say a year. And then in a year they start fruiting like this, like nobody's business. And they'll fruit like that for a couple of years. It's a really long, drawn-out process. You have to have the land available for use. Cardboard boxes. Let them run around the garden. You can grow them on an old pair of jeans. Fungi can grow just about anywhere as long as you get it some sun. And the great thing about this is that those genes are now broken down. You can throw those in the compost pile. You would never throw your genes in the compost pile before this. They would just lay there. But after they're broken down with fungi, then they're compostable. Okay, 4 principles of mushroom cultivation. Nutrient source. Inoculate the substrate. Manage the environment to favor the initial growth, and then manage the environment to favor mushroom production. This is the tough one right here. Okay, the process is a simple process. I'm not going to go into it. Well, not so simple because there's a little mycology involved, a little microbiology. I won't go into it much right here, but 7 days, 10 days, substrate preparation, spawn run, production flush for about 6 weeks. Total time, 40 to 90 days till finish. That's sort of a rundown on the production. Okay. The 3 things you have to be concerned about in mushroom production, the 3 things you have to control, and this is only during fruiting, because most of the time you can grow your mushrooms on a table here. They'll colonize the bag on, it'll colonize your substrate just sitting here on the table. many different temperatures, but it'll never fruit. But if you want to get the fruit, you have to do this. You have to, very specific, relative humidity above 85%. Temperature less than 80, preferably less than 70, and then CO2 less than 600 ppm. Why CO2? Well, you figure that mushrooms, when they grow underground, they do not want to fruit underground, except the truffle. Most mushrooms don't. They're actually, and CO2 levels underground are very high. They're actually under CO2 oppression. When they come up to the surface, wow, they get a breath of fresh air, or at least they get released from CO2 repression, and they form a mushroom. So if you want these things to fruit, you have to have your CO2 about ambient. Now, that sometimes is a challenge, because if you have a greenhouse packed with mushrooms, and these mushrooms are respiring like we do, oxygen in, CO2 out, your CO2 concentrations go up through the roof. Your CO2 is like packing all of us into a little box, and that CO2 concentration is going to be up 10,000 ppm in no time. Mushrooms will not fruit then. They will not fruit then. Okay. But for profit, and so that's how you fruit them. But really, how do you make it profitable? You have to focus on what's known as biological efficiency. And biological efficiency, and this is how we compare energy or substrate conversions. Biological efficiency, fresh weight over dry substrate weight times 100. Okay. Everything about the process can affect bioefficiency too. So you have to be careful of the process. Substrate, spawn, temperature, humidity, sometimes lighting. Sometimes mushrooms do need a little light to fruit. They're not just growing in the dark. That's misinformation. Okay, comparison of bioefficiencies. Now we can convert plant products into animal products or into other protein sources in a variety of ways. And if we take these processed pelleted feed and fed them to cows or something, you took 100 pounds of them and you gave it to cattle, you get about 5 to 15 pounds of cow, right? You did this to chickens, you get about 30 to 50 pounds of chicken. Just start with 100 pounds. Fish, you get 60 to 80 pounds. Mushrooms, though, 80 to 150 pounds of mushrooms, bioefficiency. Extremely efficient in substrate conversion into protein, into good protein, not crummy protein. Okay, and the great thing is you can feed mushrooms things that even the fish and the chickens won't eat, right? They're not too picky, right? They're not too picky. And then also, when you're finished with them, you can feed the remains of mushrooms back to the chickens and the fish. It's great. It's closing these loops. So when you get these integrated farming systems, mushrooms form a really critical part of that in converting some of these waste products back into usable substrates again, closing those food production loops, closing those energy loops. This is what fungi can do. We saw that right at the beginning, that nice little triangle, closing the loops. All right, what do I want to talk about? Total production efficiency can be increased with co-cultivation. We kind of know that. That's why we're trying to integrate many things in one place. Polyculture food systems. Really, we do this because we can get improved yields. We can get improved disease management. And there's talk about an integrated system having climate change resiliency, right? And basically, we're looking at spatial and temporal separation of resources. Here's a good example. Interestingly enough, why is Pennsylvania the center of mushroom production in the U.S.? Because in 1885, the Quakers started integrating mushroom production in their carnation and rose industries, in their greenhouses. And they wanted to utilize the space, the spatial separation of the greenhouse. And they started growing mushrooms underneath. And it slowly built an industry. There was support around that industry, all focused in there. And then it just bloomed in this little focus. Okay. And we're looking to use the same resource more than once in these integrated systems. So, for example, here you can take the soil from the spent soil from this rose production or carnation production, and it still has some nutrition in it, right? Some nutrients. You can make your compost with it, integrate it into your compost. Again, extracting it, getting more use from the same resources. And then, of course, you're going to be cycling these resources because the plants are going to be taking in CO2, releasing oxygen. The mushrooms are going to take in oxygen, releasing CO2. Again, creating that cycle. It's a really great cycle. It's a really great cycle. Okay, but perhaps where fungi really have their strength in terms of sustainable farming systems is you can recycle your waste. Like I said, mushrooms aren't too picky. They're not too picky. The greatest decomposers on earth, liberating nutrients, and I mentioned they accomplished that with this arsenal of extracellularly secreted enzymes. Let's see, you can straw, corn husk, tomato pumice, coffee pulp, lemon rind, sugarcane bagasse, right? Okay, and then the nice thing about it is this spent substrate, after some of these things you can't really, wood byproducts, you can't really compost that very well. Compost really slow. But if you throw it in and let the mushrooms colonize it first, they begin that degradative, that breakdown process, and then those things that were uncompostable now have turned into a wonderfully compostable substrate, and you can feed it back into your systems. Again, closing loops, closing loops. All right, enormous resources are lost in modern food production. Pre-harvest losses, pre-harvest losses 10 to 30, 50, post-harvest. Post-consumer losses after you buy it, things you throw out at home. Much of these losses are due to fungi rotting our food. But the irony is that fungi can then be used to recapture those lost inputs. It's a great system. It's a really great system. Okay, and that's what inspired us to start this program, the MycoCats, Recycling with Mushrooms. MycoCats was initiated in 2013. It is funded by students. The Green Fund is a student-funded program on campus that funds sustainability projects across campus to help the university reduce its carbon footprint, become a center for sustainable research, and as a model for sustainability, right? And student-funded, student-managed, because they give us, the students give us the money. It's a fee that every student has to pay. Okay? But the great thing about it is that the students manage the project, and then it's student-driven. And I can only use the money, if I'm going to pay somebody, to pay a student. So they fund it, and then they pay, it goes out. It's a great program. It's a really great program. And we are trying to take products from campus and recycle them. Let's see. Recycle them. And right now we're focused on mesquite, pizza boxes, coffee grounds. Those are things we recycle. Our facilities and our vision. Our vision is sort of the same vision as land-grant university, teaching, research, extension. Teaching we do up on campus. In the Marley building we have growing facilities on campus. Research, over on Allen Road we have a hoop house, permanently controlled hoop house, where we do research on yields, on techniques, okay? And then of course at Tucson Village Farms we have our outreach activities. Okay, we have this educational component. People come by, they can see mushroom growing in this cool solar-powered, evaporatively cooled mushroom growing shed. Okay, and it works. It works wonderfully. No energy. We have some water pumped over to it, so there's a little water pressure. But other than that, we don't turn on any energy there, and we can grow mushrooms. It's a little challenging during the summer, but during the winter, we produce lots of mushrooms. Okay, our partners, our substrates. We get substrates. Facility services, Desert Harvests, Leaf, which is linking Arizona edible forests, provide mesquite pods, local coffee houses, provide coffee grounds, and of course the dorms provide us unlimited supply of pizza boxes. We can do that, right? We're also exploring the use of wax paper cups from the sporting events on campus, which fill up dumpsters and dumpsters. All these wax paper, post them. You can't compost them. We sell mushrooms to dining services. They will take $10,000 a year of mushrooms from us, and we sell that. We haven't quite hit our $10,000 mark. We're trying. They'll say we'll take up to $30,000 a year if we can provide a diversity of mushrooms. Okay. We also provide spawn or sell spawn, not provide it, sell spawn local mushroom growers, and we sell colonized bags to mushroom growers as well for this trouble-free production. And our waste, again, our spent waste is repurposed via the compost cats, and it goes back into campus garden, again, closing energy loops. It's a very, very cool system, and the students run it. They do a great job. My job is just to sort of keep them going now and then, and help them with some of the vision. Okay, and this also supports applied and basic research in bioremediation. Fungi are have incredible potential for bioremediation. They can detoxify a variety of substances, pesticides, petroleum hydrocarbons, dioxins, right? Plastic degraded fungi from the Amazon. They've recently discovered a filamentous fungus that can degrade polyurethane, and it can do it under almost anaerobic conditions. It can degrade plastics down at the bottom of landfills. That's huge. That's huge. Okay? Bioabsorption, volatilization of heavy metals. Think about this, the use of this in mine tailing runoffs. Remediation of soil and water through mycofiltration, right? Our sewage, we can remediate our sewage through microfiltration processes, for pretty elaborate processes. And so in addition to increasing the production of food, we can actually increase increase our availability of arable land. We can take soil that's contaminated and decontaminate through these microfiltration processes. Mycoremediation, right? Huge potential. All right. So we do some research on this. Real quickly, I want to mention a project that Lauren Jackson, one of our graduate students, just finished up in my lab, did. And it's just excellent work, okay? We know that fungi can degrade things through their extracellularly secreted enzymes. Key among these are lactases, which are the darling enzymes found in insects, but perhaps best studied in fungi because fungi make such a great model for studying these things. And lactases are these multi-copper oxidases that are just powerful. So powerful that they're most efficient in degrading lignin, which is probably the most recalcitrant natural molecule on Earth, right? What's the most common natural macromolecule on Earth? Cellulose, the most common. The second most common macromolecule on Earth, lignin, and almost nothing can break down lignin, which is a good thing because it helps build our soils. But a few fungi, white rot decay fungi, can break down lignin, and they can also break, because of these enzymes, they can break down really nasty stuff, such as aflatoxin, probably the most naturally potent carcinogen, natural carcinogen known. Many other toxic metabolites as well. Okay, so Lauren took this project, and he wanted to look at oyster mushroom production. He wanted to look at oyster mushroom production on natural products around Arizona, such as growing on mesquite pods, right? Mesquite. Comparing bioefficiency of mesquite pods supporting mushroom fruiting, compare the lactases from different fungi, compare them to oyster mushrooms and different strains of oyster mushrooms, determine the optimal condition of solid state fermentation that's ramifying through the substrate in which this degradation of aflatoxin occurs, determine the suitability of the feedstuffs after they've been colonized by oyster mushrooms for use as feedstuffs. So once things are contaminated with aflatoxin, you cannot use them. You can't even feed them to your animals. You have to throw it away. You have to throw it away. And determine whether the mushrooms grown on this contaminated feedstuff are free of aflatoxin, other organic toxins. And I'm not going to go through this because I'm running a little late. Yield and nutrition responses of Pleurotus austratus cultivated on mesquite pods. Interesting, we looked at the bioefficiency of the, looking just at the proportion of wheat straw, the proportion of legume beans, the proportion of mesquite wood. And we found that varying the amount of straw, if you looked at just the straw, did not significantly affect the bioefficiency. But if we look at incorporating increasing amounts of mesquite pods, our bioefficiency was significantly increased, right? To 100%. Increasing the amount of mesquite wood, because we were looking at different mesquite products, increasing the amount of mesquite wood decreased the bioefficiency. This is a wood decay fungus. We did not expect this, but so we learned a little bit about oyster mushrooms, but we kind of learned a lot about mesquite wood and its suitability as a fungal substrate. Mesquite must have some really strange things in it that the mushroom does not like, unlike maybe oak or cottonwood, which these fungi really love. Okay, he looked at the ability of the fungus. Once you took these contaminated products, actually we were looking at corn here, contaminated corn, ramifying it with oyster mushrooms, letting the oyster mushrooms colonize it, and then look at the look at the aflatoxin content of that corn after 4 weeks of colonization, and then looking at the mushrooms afterwards as well. And we found in this study, let's see, up at 2,500 parts per billion aflatoxin, corn contaminated with 2,500 parts per billion aflatoxin, and this is without colonization. And after 4 weeks, this is what we found. The aflatoxin levels dropped down to less than 250 parts per billion, from 2,500 parts per billion. Now, we can't do anything with this. We can't do anything with this. But this, we can't eat it still. We can't eat that corn, but we can feed it to livestock for final finishing on livestock, which I think the U.S. level is at 300 parts per billion. You can feed that aflatoxin contaminated livestock to cattle for finishing up, not to dairy cattle because it gets in the milk, but for cattle. So you can take this stuff that was trash, heavily contaminated, and turn it into another usable substrate. And then also the mushrooms, whoops, uh-oh, interesting. Well, that's kind of strange. All right, conclusion. The conclusion was, if I remember correctly, the conclusion was that the mushrooms grown on this aflatoxin contaminated corn was undetectable. The mushrooms grown on it. The corn itself dropped not over 90% aflatoxin contaminated, turning an unusable substrate into a substrate that you could feed to other livestock, use other purposes as well. And there was some other, let's see, oh, here we go. This was, yeah, Lauren's slides. Okay, I'm going to just go through this. Okay, so minimal reversions, because sometimes when you degrade some toxins, they can revert back. Minimal reversions, reduce mutagenicity of the things that were remaining in the substrate. It's cheap, it's practical. It's not so simple because you have to use, you have to be able to grow the fungus. And it's not necessarily perfect because it doesn't take it to zero, but it does a pretty good job, okay? All right, let's see. Okay, so I'm going to finish up here. That was just some of the research we're doing. I have another student who's working on using the oyster mushrooms and specifically the laccases produced by the oyster mushrooms in biological fuel cells. It is some really cool work. Okay? So we have all these spin-off opportunities from studying mycoculture, including integrated into our sustainable system. Mushroom production is an incredibly diverse, flexible, sustainable, and profitable industry, right? That can be integrated in many sorts of cropping systems. So, of course, this question is to Gene, right? The question is, where will we grow mushrooms in the future? Aha! We can grow them underwater, right? Off on the. So limestone caves are kind of, rare, far and few between. We have a whole continental shelf all around our continents. We have lake beds, things that we can put underwater farming. It's a growing interest, right? Underwater farming. You don't need light. And it's thermally stable, or pretty stable, right? It's like a limestone cave underwater, right? Or we could take these lunar modules. And so my challenge to Gene is, when are we going to grow mushrooms in space, right? When are we going to grow them in space? All right, very good, very good. So my plan is, if you want to live to be 100, eat your mushrooms and enjoy. Okay? And I'll take questions. And I was supposed to actually ask you to interrupt me repeatedly with questions during this.
We tried to interrupt you. It was impossible to interrupt you.
I'm sorry.
Where do you get all your energy? It's great.
All right.
And you will definitely live to 100. We have a question here.
Hi. You answered my question. Answered my question sort of with one of your last slides about where the applications will be, and space is one of them. And I guess I'm the only one here from the Tucson L5 Space Society, probably because of the time. At the very beginning, there was
at the where?
At the very beginning, there was a mention made of it's being haploid for most of its life, as I guess as opposed to diploid,
which we are in most
of it. So could you, if that's in your
okay, so we have you're interested in the biology of fungi. That's really
that's one of the things.
Okay, so they're haploid for most of their life, but they have to reproduce. So the main, for us, the main, what are we, the gametophyte or the sporophyte? We must be the sporophyte, and our main, our life cycle is primarily existing as a diploid. But when we engage in sexual recombination, we have to make gametes with our haploid. So part of our life cycle is haploid. It's very short-lived, just our gametes. And then we quickly revert back to a diploid condition. Fungi do it oppositely. They live most of their life as a haploid. And then they quickly will form, they'll form some gametes that are also haploid. They'll come together very quickly as a diploid, and then quickly meiosis occurs and they go back into being haploid again, and that forms their life cycle. It's very odd.
Can that be manipulated to be of even more benefit?
Wow. I hadn't, that's a very good question. Can it be manipulated to be of more benefit? I don't know if I'd have the answer. Just, I might have to have a glass of wine and some chocolate with Dr. Wells' food pyramid to come up with that answer.
Because if you're stuck in space, for a long time.
Right. Oh, very interesting. Very good.
I don't want to monopolize.
Very good.
Other questions? Over here.
What about pests? I've
very
gone
good.
out hunting king bullets, and the worms got to before I did.
Very good. So you know, I'm a plant pathologist, so I'm worried about diseases of plants, but there's a whole field, and plant diseases are caused by fungi, but then of course fungi get diseases too, and they get bacterial diseases, they'll get viral diseases, and they'll get other fungal diseases. And there's also big pest management as well. We learned a hard lesson, or we're learning lessons, not hard lessons. We're learning lessons over in our hoop house over on Allen Road about pest management in our mushrooms. Oh yes, so we get one of the big problems with commercial mushroom production is you can get fungus gnat contamination. Okay, and they get in the bags, particularly if you have open bed cultivation. But even if you have cultivation in bags, and once you open up the bags, those fungus gnats just go woo, and they go right to it, right? Start reproducing there, and it can reduce your yield. So there are, and you can grow mushrooms organically or non-organically, and there are treatments, insecticides, fungicides that you can use for your mushrooms as well. An interesting pest problem that we had, which I never really thought of before or heard of before, over in our hoop house is our mushrooms down at the very bottom shelves in our hoop house. And our hoop house has gravel on the bottom, and it's kind of moist in there. And I was harvesting mushrooms, and I picked some up, and I looked at the bottom, and the gills, I said, what's going on? It looked like somebody peppered the gills, little tiny pepper. And of course, I couldn't really see what was going on, so I put on my glasses, and I looked very closely, and the little pepper things were moving. What the heck? Pounded them off on a piece of paper, wrapped it up, took it into the lab, looked at them. They were, oh my gosh, now I can't remember. They were springtails, little tiny springtails. They are not problematic. They don't drill into mushrooms, but they're cosmetically unattractive. So people would not want to buy a mushroom and look at the gills and see little black specks moving around. It's a little springtail, so it was a challenge. We had to clean out our mushroom house.
Just for clarification, this hoop house you're talking about, that's a controlled environment greenhouse.
Correct.
Okay.
Absolutely, Gene.
Make sure everybody out there understands that.
It's a hoop house, a controlled environment greenhouse, and it works very well. It has a big evaporative cooler on one side. It has a big evaporative cooler on one side. We can keep in most of the year we can keep the temperatures below 70 degrees in there. With this, the whole back wall is one big wet wall. And of course we have fans blowing in there. In the summer we're challenged, especially when the monsoon comes. We have a challenge keeping it below 80 degrees. Now we could keep it colder. We could keep it cooler, below 80 degrees. The problem is, and the way you would keep a hoop house, a controlled environment hoop house, cooler. I learned my lesson. I'm going to cooler is you turn on the fans. You turn on the fans and you pull more air through it as much as you can. What happens then? Well, we had three considerations. We have humidity, so we're still okay. You have fresh air, you have temperature, and humidity. When you start pulling the air through the evaporative cooler, you really start doing air exchange very rapidly, and our humidity dives down. We have to supplement our humidity in the controlled environment hoop house with these with these big cans, water containers with atomizers in them, and it makes a fog, kind of like a dry ice fog, very fine mist, and it floats throughout.
There was a question online about where to get the spores.
The spores. Oh, you can go online and buy mushroom spores. All kinds of mushroom spores, actually, from around the world. You know, especially the Netherlands, you'll get the really interesting mushroom spores. But you can. There's a lot of companies in the U.S. that support the mushroom industry. You can buy spores. You can buy cultures. Most people buy cultures. They don't mess with the spores. They buy the cultures. And you can buy colonized substrates that you can packages that you can just slice and fruit by yourself. You know, to encourage people to appreciate, enjoy, and become interested in mycology and mushrooms. So you can get these all online. Let's go. And you'll get a kick out of the Dutch sites too. Wow, very interesting.
There's another question online from Obregon, Sonora.
Okay.
What specific conditions do you use at the U of A in Tucson to grow fungi?
Okay. So we grow our mushrooms on several different substrates. Our most common, our fundamental substrate is based upon straw. Straw is cheap. Okay. And we modify the straw with either mesquite pods. We can do it with cottonseed or cottonseed meal. Or we can go to the feed store and buy alfalfa pellets and mix it in. And we do various combinations. Right now we're running at about 50-50, 50% straw, 50% of these other things. We think it's a little bit too rich So we're moving it down. Hopefully we're going to get down to about maybe 20% say mesquite pods and 80% straw. And then you have, again, you can, once you colonize that substrate, and we sterilize our substrate, so this is a whole nother seminar. You can sterilize that substrate and then inoculate it and let the fungus ramify through it. And then comes the challenge, fruiting it, which you need those conditions. Less or greater than 85% relative humidity, less than 80% better, less than 70% or 70 degrees centigrade, and then less than 600 parts per million CO2. Now we put all of our mushrooms once in one of these incubators over here at the SEAC facility, and we were doing our what's called the spawn run, colonize our substrate in this room with a big sealed door, and we'd open up. It was 20,000 parts per billion, parts per billion CO2, parts per ppm, parts per million, I guess, in CO2. Yes. Oh, I'm sorry. Oh, 70 Fahrenheit. I'm sorry. I'm sorry. I'm sorry. Yes, that is boiling. I'm sorry. 70 Fahrenheit.
I'll give you one of the areas that you've touched on, and in the human cancer side from epidemiology, the aflatoxin has been associated with rotten peanuts, and the aflatoxin in Africa that was relatively specific to hepatic, i.e. liver cancers.
Bad. Too, is on
that sounds like an area for which there would be wonderful opportunities here to get into the cellular biology of how is this really happening and where is that interface. And as you probably know, Lipitor and the other anti-cholesterol drugs largely work through metabolism in the liver. So,
Right.
So you've got a wonderful interface here to work
Well,
with.
so back here, one of Lauren's, okay, this was interesting. The International Journal of Veterinary Medicine, hepatic protection effects of oyster mushroom consumption on broilers fed aflatoxin. So even that detoxification can occur within us, within the animal system. So that's very cool. That's very cool. Now, there's other ways people have looked at decontaminating feeds from aflatoxin. They use an ammoniation process. There's one or two other processes. Lauren knows all about this. Oh my gosh. You know, the students always know more than the professors.
Other comments?
When you spoke to about the commercialization of and marketing of mushrooms, can you talk a little bit about post-harvest handling and shelf life? I mean, you talked about shipping them everywhere. And
very good. That's a good point. Now, and I got to also sort of clarify that I've only been doing this about 2.5 years, so I've been on a steep learning curve about this. But it is a very important aspect of mushroom marketing. The button mushroom, none of us could really probably break into the button mushroom market. That's dominated by big players in Pennsylvania and California. Monterey mushrooms. Oh my gosh. They produce so many button mushrooms, and they've got the price down so low. They run a very thin profit margin, right? They can't mess up their processor. Those mushrooms, the reason why button mushrooms are so have such utility in terms of a product to produce is their shelf life is very long. It's 2-3 weeks. You know, you can ship them anywhere. The gourmet mushrooms have a much smaller or much shorter shelf life. The quality of an oyster mushroom freshly harvested versus one that's 3 days old can be quite dramatic, or 8 days old. You know, sometimes when you get these oysters in whether it's in Whole Foods or Bashas', they're all beaten up and broken in bits and pieces. I mean, it's really, you say, oh, what is that? Oh, that's a mushroom. But you know, but if you get them within a couple days, they're wonderful, absolutely wonderful. So there lies the opportunity for local mushroom production. The quality difference between something that's grown in Tucson for a consumer in Tucson versus one that's grown in Pennsylvania for a consumer in Tucson is huge, a huge difference. And that's why some of these large companies have not jumped both feet forward into this gourmet mushroom market, because they know they're going to be challenged unless they diversify their operation and put little Monterey mushrooms everywhere around the country, little Monterey mushroom companies all around the country, and really focus on those local markets. And you know, we have this whole sustainability movement right now at eating local and trying to get our food within what, that 250-mile radius. And this fits in perfect with that, because by eating mushrooms that have been grown within 250 miles of your home, you're guaranteed, or almost guaranteed, a really high-quality product, really high quality.
There's another question from online that,
Okay.
is something I'm interested in. We were discussing a little bit about the bioregenerative life support systems that we're working here to keep people alive on another planet, using plants to produce oxygen and sharing the CO2. What I've been hearing here is that a mushroom is much like a person or a human,
Yes.
and we'll have to share the oxygen with mushrooms, or will the mushrooms take us over so that they can have all the oxygen?
So you mentioned this when I said, oh, we're going to grow, and you looked at me and said, Barry, we'd like to reserve the oxygen for the astronauts.
Amen.
You know? Okay, so the vision is this. When you have a garden up there in space, and it's orbiting Europa, for example, supplying our colonists on Europa, you're not going to have people in that greenhouse. It's all going to be robots anyways. Who cares about whether there's oxygen for people anyway? It's all going to be robots. You know, you don't have to think about it. The astronauts aren't going to be flying through space with their little garden behind their seat, you know, doing that. No. It's all going to be robots. Our greenhouses are going to be...
On a habitat, too, it would be the same thing.
Yes. Our greenhouses are in space. My vision, which is kind of odd at sometimes, but my vision is that we'll have greenhouses, you know, as we move out into the universe, out to the solar system, we'll place stations out there that'll supply our ships at different points. And nobody's going to be on them. They'll be self-running, automated stations, growing food, packaging their own food, sitting aside so that the spaceship comes along, docks up and says, pulls off a pallet of food and takes off. Doesn't probably even, okay, that's sci-fi.
You're getting there. Understand.
I'm almost there, Gene.
Yes.
I'm
You're
almost there.
Okay. All right, another comment.
Thank you very much for your presentation.
You're welcome.
In my country, actually, this is a very profitable business. I mean, if they consider it. Our people like to eat truffles or turf. I forget truffles. Truffles, or turfes, or I forget the name actually.
Okay.
So truffles. Actually, it's almost 1 kilo or 1.5 kilo can be with 100 or $150. So sometimes that's gonna be if they think about it as a commercial
opportunity.
Yeah, it's gonna be very profitable.
Even though nobody in the neighborhoods can afford it. Oh no, I'm sorry. You had more? Oh yes. So
it's a comment, not a question.
A comment. Yes, it's very profitable. And in terms of exports and truffles are pretty durable too. They're tough. Yes.
We're going to have to wrap this up.
All right. This has been great.
Well, first of all, I think it'd be a shame if we didn't have people in space growing things.
Or on the continental shelf.
Yeah, well, a lot of places, but you know, I think it's part of humanity's role is to take plants and other creatures to other places, you know. But aside from that, I'm interested in the whole problem of waste recycling and the potential of using mushrooms for recycling waste, particularly food waste and maybe weeds and that kind of thing. And I'm wondering what sort of treatment of the substrate would be necessary, or could we just, you know, do windrows or some sort of simple system to accelerate the process and then use the finished material and compost or something?
Okay, so that's a good question, and I'd have to ask the question associated with, is the purpose of our activity to recycle or to make a profit? Because there will be some intersections of objectives there. I mentioned that the whole mushroom cultivation, we're all interested in bioefficiency. Bioefficiency, we have to keep maximum bioefficiency. Bioefficiency is impacted by your growing conditions and by your substrates. So we may be able to recycle a lot of different things, lawn clippings even. You know, you'd want to dry them down. You want to dry down everything in your yard or whatever, and probably make little bales and dry them out, and then sort of prepare it properly for mushroom growth. But if you wanted to do it for profit also, you might not get the bioefficiency out of some substrates that you would out of others. We've done a study where we did look at, I mentioned three things. We looked at mesquite pods, cottonseed, and alfalfa pellets. And our hope was that mesquite pods was going to give the best bioefficiency. That would have just been great. We have mesquite pods raining down in Sonoran Desert. Oh my gosh. And
there was a question online, is that with or without the seeds? Just the pods themselves? Or
the pods, no, we just chipped the whole thing.
That answers the question now.
Yeah, including the seeds. And it turned out, and it was a significant difference between the three. Cottonseed was the best, followed by alfalfa pellets, followed by mesquite pods. Dang it. We really wanted the mesquite pods to be the best because it would have been a perfect silver lining then. There. The best mushrooms produced, the most flavorful, the most amazingly delicious and fragrant mushrooms were from the mesquite pods. You grow mushrooms from mesquite pods, wow, they have a flavor, a smell unlike anything else. It smells like garlic and black pepper all mixed together. We started growing them, we did it several years ago and I noticed it, but we've kind of been focused on cottonseed lately. And we said, okay, let's start up now that we have more mesquite pods this year, we'll start up our mesquite pod production again. And immediately I noticed it in our hoop house controlled environment facility. and I walked right in there. I opened the door and I went, oh my gosh, we're using mesquite pods again. I walked in there, you smell the mushrooms, you go, that is amazing, that is wonderful smelling. And you wouldn't think that. Mesquite pods, garlic, black pepper, how did that happen? Well, compounds get broken down, new it's amazing.
We have the same opportunities with the tomatoes that we grow, so
yeah.
it happens. We're going to have to call this to an end, and Rafi's going to give some thanks here for you being here. But those online, I want to thank them for all the great questions. We didn't get to all of them, but we got to a number of them. Rafi, please.
I just wanted to say that when I identified Barry as an inaugural speaker, I thought it was going to be a good seminar. I was sitting here breathless. I think you presented a wonderful, wonderful story. And who would have the lowly mushroom can be such an amazing organism? So thank you very much for educating us. Thank you for being here. And thank all of you. And Dave Bodnar, thank you for running that show.
Okay. Thanks.